Recording pending. Never seen that before. Okay, that worked. Great. All right, so today we are going over Chapter 2 in the lecture. Also, hopefully you've already read through all of Chapter 2 at this point, because this evening, just before midnight, your Smart Book for Chapter 2 is due. So just wanted to remind you of that. Then we're going to have a Smart Book of Chapter 3 right after the weekend. So get done with that or read that over the weekend so you can complete that on Tuesday. And then the last chapter for our unit will be Chapter 4, and our first exam will be coming up at the beginning of February 5th and 6th. So I wanted to give you a little more information about that so you can start to study for it as we are going through. Oh no, one of my videos has disappeared. I do need to log in, and I'll tell you how many minutes you'll have for the exam, things like that, some study tips. Hopefully right now, let me know if it's not showing up, but you should see on your own Canvas the Exam 1 study guide. So you can download that right now, start working on that, answer the questions relevant to Chapter 1 and then Chapter 2, and just kind of use that as a study tool. Or you can download it right before the exam and in your study sessions right before the exam too. But you should have access to that currently right now. So Unit Exam 1, it's 90-minute time limit for that. All of the exams are multiple choice in this class. I'll check how many questions are on this, but I actually have to move it over here so you can't see all of the questions. So it will be 60 questions, and you will have 90 minutes to take it. It will be open book and open notes. So what I hope that you do, this is basically good study practice for any class, not just this class. Cognitive psychologists have studying to see like how best to study, to take tests, to learn things, to do well, to get it into your memory. So some tips that I want to give you are, one, as you read through a chapter before you talk about it in a class. For this class, for any class, so read over chapters 1, 2, 3, and 4 before we talk about it here. And as you do that, take notes. So make the notes meaningful to you. So examples that you think of in your own life, examples from the textbook that were relevant to you or that made you understand it well. So put those down in your notes. Then when you come to a lecture, add to those notes. So you can do that a lot of different ways. Just have a lecture notes page or just add it to the same space in a notebook or something like that. So however you want to do that is fine. But adding to your notes from examples that I give in lecture. For example, today we will watch some videos on epigenetics that go into a lot more detail and a lot more examples than your book did. So if those are helpful to you, you can write those examples down, or a way that I describe epigenetics that made sense to you. So then you've experienced the material from the chapter, from lecture, and then you have your notes. Then you can review your notes before an exam. So reviewing those notes and then rewriting your notes is actually a really good thing. So one thing I recommend for all my exams is actually that you make a note card. So it doesn't have to be that specific size, but to make a note card of material that you've been having trouble remembering as you review your notes. So if you're reading through your notes or rereading through the chapter or something and you say, oh, I don't remember this at all, maybe putting that on a note card, because that process of rereading it and putting it into notes or rewriting your notes onto a different page, that does a lot for your memory to get that material stuck into your memory. And then when you go to take the exam, even though it's open note and open book, you won't even need to reference your notes very much if you've gone through all those steps, because you'll remember so much of it already. And there might be a handful of questions that you're feeling unsure about or don't remember at all that then you could go back to your notes or back to your book for. Because with that time limit of 90 minutes and having 60 questions, that's not going to be enough time to look up the answers to every single question just going into an exam cold. You'll definitely need to have studied and read and understood the material beforehand. Another thing I wanted to let you know, for the exams, I like to have most of the questions be reasonably difficult, like not too hard, not too easy, but then I like to have about a tenth of them be really easy questions. So if you just barely paid attention at all, you would get those right because they're just so easy. And then about 1/10 of the question to maybe 15% of the questions be really difficult. So 10 to 15% really easy questions, 10 to 15% really difficult. So reasonably difficult would be like the example questions in the chapter, if you've ever looked over those. Some of them are a little difficult, some of them are pretty easy. That's about what most of the exam is. And then about 10 to 15% of the exam's really easy, and 10 to 15% of it is pretty difficult, where you need a good understanding of the concepts to understand what the question is asking to then answer it correctly. So that is some recommendations. Also, you have the study guide. You have access to it now. It has some example questions. Actually, I can show it to you. That might be more helpful. Since we're talking about the exam right now, let's open this up. Let's just open in Firefox. All right, so these first questions in the study guide, again, you don't have to start with the study guide right now, but it's available to you if you want to get started. So these open-ended questions are basically questions that you would find a few multiple choice questions on the exam. So topics that there will be more than one question on. Then down here, you've got multiple choice questions. You'll see either these exact same questions or one very similar to them on the exam. So a synapse is a blank. So reading through those, so from 10 to 14 or 10 to 18. Wow, this is a really long study guide. So all of those questions are essentially exactly what you'll see on the exam. The same kind of format, same kind of questions. I generally try to put a couple of the more difficult questions in the study guide so that you're prepared and see it before you see it on an exam. So are there any questions logistically about the exam? I know it's still a little bit away, but I do want you to have these ideas in your mind of how to prepare so you can start your study process, or if you haven't been reading the chapters, you start doing that now. All right, I don't see any hands up, so if you do have any questions, definitely let me know. Again, the exam will open on February 5th and close on the end of day February 6th. So opens basically right when February 5th starts and closes just before midnight on February 6th. All right, so let's jump into the lecture for today on Chapter 2. So we finished talking about a lot of the basics. Now we'll talk about some of the basics of genetics, because genetics is kind of that nature part of that, yeah, the nature part of the nature-nurture argument. A lot of that comes down to genetics. So let's talk about natural selection. Hopefully you've been exposed to this in other classes, so we're not going into terrible detail. But essentially, natural selection and adaptation is essentially the idea that any species, any kind of feature or ability or feature like a beak size or shape that is adaptive for keeping that individual organism alive will have a higher likelihood of being passed down to its posterity. So if you take a big family of birds, so let's say you have 200 birds on an island and you split them up into 2 different islands. The birds on each island after the course of a couple hundred or a couple 100,000 years might look very dramatically different to each other, because as these birds have babies, then the genes and that process of mixing genes together, mutations can come in. And so with those mutations, it might be a slightly longer beak or a slightly harder beak or a slightly different wing shape or a slightly different color. And so if that slight change or variation is adaptive for that individual bird, it might get passed on to its children. And if it's still adapted for those children, it might be more likely to pass on that trait. So those slight mutations can then lead to an entire species over the course of generations kind of adopting that same trait. So those 2 families of birds that we split between 2 different islands, 100,000 years, well, maybe not even that long, a couple thousand years later, after multiple generations, we might see them as very, very different birds. They might look to us very different, even though their common ancestor came from the same family of birds on one island. So those adaptations, they come from genetic mutations. This is unfortunately not like you see in the X-Men. We don't get mutations of like weather controlling powers or like laser eyes, although that would be really neat, probably a little dangerous. But you could have something like a genetic tendency to have slightly more white blood cells or something like that. So maybe you heal from wounds a little bit faster, not quite like Wolverine, but maybe a little bit faster. So if your ancestors had that trait, or if our human ancestors had that trait, their posterity might be more likely to live, be able to fight infections, things like that. So then now it kind of spreads out through the human family. So that is how adaptations happen. They come through slight mutations in the genetic code, not big dramatic changes like lightning powers or laser eyes, which is a little unfortunate. But if you want more detail on this process of natural selection, this is a good video for it. But hopefully these are concepts you've already encountered in the past. So what do evolutionary psychologists do? Well, they look at that process of adaptation and they try to use that to explain any kind of human behavior we have. So how does our memory work? Well, an evolutionary psychologist would say, well, we probably evolved the memory capacities in XYZ ways, and try and look at that. Try and look at other relations to humans. So maybe look in apes or mice and see how they evolved, see how that was different than how humans evolved or something like that, to be able to explain human behavior, human cognition, human emotion from the idea of evolution and those adaptations. Now, some things in human life come very much from that nature side, that genetic side. Some things come very much from the nurture side. And so finding that balance takes a lot of study, and we'll talk about a couple ways we do that study. With that, a lot of human development, a lot of human behavior has come because of adaptive behavior. So for example, the reason we like to come together as groups, that we're very social creatures, is because it's been evolutionarily adaptive. We raise young together as groups. We are adapted to do that. We learned how to hunt together, things like that. If human evolution took a different path, if we were all living as just individuals, our society would look incredibly different than it does today. But that working together has really impacted how groups and how humans can form. Take, for example, cats. So a baby cat is very small and very helpless for a very short time compared to a human. So a 4-year-old cat is completely independent, has been an adult, has maybe even had children of its own. My 4-year-old child, if I put out to survive in the wild, would not, right? So we need to care for our young as humans for a lot longer period of time. They require a lot more attention. And part of that is actually down to brain development. So converging paths of evolution led for things like the ability to grow bigger brains as we got into groups. And so as a lot of these features have led humans to do the behaviors we do now. But again, not everything is exclusively dependent on behavioral genetics. So, and as well, some of those adaptations are not necessarily adaptive to us now. Some good examples of this are that our human ancestors lived in food scarce environments. So we have evolved, humans have evolved the capacity or the genetic tendency to like high fat and high calorie foods. Because when those became available, storing up on them, eating a lot of it was good for our survival. But now, in places where we don't have a food scarce environment, where it's maybe a little easier or cheaper to get a hamburger versus a salad, that's not necessarily good for obesity, right? So this adaptation is not necessarily adaptive to the environment that we have built as humans now. Another good example is our stress response. So I wonder how many of you, maybe just a show of hands, using the raise hand button. How many of you saw the video, I think it was like 2, maybe 3 months ago, of the person, I think it was Provo Canyon, that was being followed by a cougar? So I see Mackie is raising his hand. We've got another hand raised. Okay, 3 of us, well, 4, 3 students and me. All right, if you haven't seen it, I mean, it doesn't really help too much since we're going to talk about the example right now, but it's a really interesting video to watch, so I definitely recommend doing that. So in that situation, this person was hiking on their own. They encountered a cougar, and essentially they walked backwards facing the cougar for, I think it was like 7 minutes. So this whole encounter with the cougar lasted like 7 minutes, where they were stressed out. So a big cat following you, they had a lot of stress response. So that's essentially the exact situation we have evolved to handle. So at the end of the video, you can hear the person, like after the cougar's gone, kind of laughing a little bit hysterically, breathing heavily, those kinds of things. Like you can tell they're starting to calm down, but their stress response is still high. So that's what we've evolved to do, is that fight or flight. So quick, immediate stress, fix the stress, run away, kill the thing, whatever it is. So that's our evolved capacity. We've evolved a stress response in that way. So that stress response is not very adaptive to today's society, where we have a test coming up in a couple weeks. Maybe that's stressful to you. You have a rent to pay in a couple weeks. That's stressful for you. I'd never like to mention it, but student loans might be a stress for you as well. It's definitely for me, and it's very triggering to me, that's for sure. So those kind of long-term stresses, maybe you've got a big project at work that the deadline's a couple months away, but you have to work on it constantly, and so you're always stressed about it. That is not how we are built to handle stress. We're built to handle that quick, immediate stress, get away from the cougar or kill the cougar, and then calm down. But when that stress is over a very long period of time, take, for example, being in poverty and having those additional stresses all the time, we have not evolved the capacity to handle that kind of stress. That's not what our experience has been. So we're not evolved or adapted to it. So our stress response can actually be pretty bad in that kind of situation, lead to things like anxiety disorders. So an example of how our evolutionary adaptive capacities in certain contexts are not so adaptive. So with that, while we have these physical structures, the brain structure, and these propensities in our biology, it is not the end-all be-all of all of our behavior, all of our decisions. So it's not all exclusively determined by genetics. So Tamara, you've got your hand up still. Is that a question, or was that for the cougar? I know you put it up for the cougar. Okay, it's down now, so I assume it was just the cougar. I just didn't want to pass over you if you did still have a question, so sorry about that. All right, so what are genes and what are they doing in our body? So if you've taken a biology class at the college level, you've gone into this in a lot more detail than we're going to cover right now. I've got a couple of resources to find more information, but I'm going to cover it at a really basic level. I know for some students it helps to have a little more of the complexity, a little more information about the chemical processes and biological processes that are happening. But for some students that just is confusing. So I've taken the road of doing really basic here. So a chromosome, hopefully you've heard of chromosome before at least, is a thread-like structure in every single human cell made of DNA. So inside each of your cell's nucleuses, inside the nucleus of each cell is your chromosomes. So that chromosome is basically the wound up bits of DNA or wound up strands of DNA. So what is DNA? It's a molecule. It's a really long molecule. It's a really complex molecule, but that's all it is. It's a specific molecule made up of a couple different chemicals. So they're termed or they're labeled ATCG. So those each have chemical names, but for our purposes, that's all you really need to know. So the structure of that molecule, the order of the ATCG, so I'm imagining my fingertips are like the bits of genetic information, so the ATCG and then ATCG. How those are organized, so if they're organized like this way, so it's ATACG, so that order determines what the gene is read and then does. So the DNA is just that long complex molecule. It's the same throughout your whole body. So each cell has a nucleus and each cell has that genetic information. What those genes are is the bit of that DNA that kind of gives instructions to your cell of what to do. So it tells cells how to develop, which proteins to create. So for example, a cell for your muscles versus for your skin, it's the DNA in both of those is the same. But for your muscles, it's read in a specific way that it then becomes a muscle cell. For your skin, it becomes a skin cell. And we'll go in a little more detail of that in a second. So what is a protein? Essentially, it's the building block of cells. The DNA itself is actually wound around specific proteins, which is just another chemical. But also proteins can come and read from that order. So the ATCG, the protein can come and read what that order is, and then it tells that protein, hey, go and move energy or go move this chemical from this place in the cell to this place in the cell. Or grow in this way, or do this kind of thing. So the proteins read the genetic instructions from the DNA. So the DNA is like the long strand, it's like the instructions. A protein comes and reads a portion of that, and that's what we call a gene. And when that protein reads that section, it goes off and does something in the cell. So we've got the DNA, this long complex molecule. The protein comes, it reads the order of the molecule, and then it goes and does something in the cell. So there's tons of different genes. When I say tons, I mean billions of different genes that you can read from your DNA. The protein does a specific thing in the cell. Maybe it grows, let's say this is a neuron. Maybe it grows a new dendrite. Maybe it moves chemicals from here to here, keeping the cell alive, those kinds of things. So those cells then make up our bodies. So that basic structure of proteins reading the genetic code and then doing things is basically what leads us to have muscle cells versus skin cells versus brain cells. And then it also is in charge of a lot of what those do throughout life too. So that's a really basic overview of like how a gene does something in your body. Basically, the gene is the instructions, the protein reads it and then goes and does something. So when we're looking at genetics in the context of psychology, a lot of times we try to determine, well, how much of this behavior or this feature or this aspect of development is related to the genes or the nature versus nurture or the environment. So one way we do that is through twin studies and adoption studies. So from your reading, what do we do with twin and adoption studies? What are we trying to compare to determine that nature versus nurture? What might you be looking for? So feel free to either raise your hand and comment with your microphone, or you can also just type out a response as well. So another way to phrase the question could also be, what might you look for? What might be a specific example that you would look for between twins to see if something's more nature versus nurture? All right, we've got a good comment here. So Rachel says, with twins, it's easier to see what's the same because of genetic predisposition and how each might be different because of the different environments they spend their time in. Yeah, very good. So the genetics of a identical twin are exactly the same, or at least close enough to be called exactly the same. So a fraternal twin, a lot of the genes, not as many as not all of them like with identical twins, but even a fraternal twin, a lot of the genetics are still the same, more than it is with just siblings. So you can look at that and say, okay, so what things are the same between these two twins? And if there is something the same, maybe a behavior or a personality trait or a physical characteristic is the same, you might determine then that maybe that's exclusively or at least more from that genetic component. Versus if there's something different between the two of them, maybe that is coming more from the nurture part, the environments that they're in. So really good, Rachel. I actually had a twin in my earlier class who mentioned that one characteristic that was the same between them and their twin was they had a crooked tooth. So their twin actually, their tooth started becoming crooked earlier on in life, but then their tooth also started becoming crooked. I think it sounded like a year or two after their twin had it. So that was probably mostly determined by genetics. But they also mentioned that their taste in guys to date was very different. And so this student assumed that that was probably more from environmental, from their own experiences and things like that. So that's kind of what the researchers do as well with twin studies, is like, okay, so that trait is the same between these twins, probably that is more likely coming from heredity or from those genes. Adoption studies are similar. You can compare the adopted child to their adoptive parents, see what characteristics or traits or capacities they have that are the same to the adoptive parents. Since they share no genetic material, that might suggest that those shared things between the adoptive parents and the adopted child might be from nurture, from the environment they're in, versus any characteristics that are still the same as the biological parents, that might be a nature, the genetic factors. So those are some of the ways that we've done this. More recently, we can map in the entire human genome. We can look at people's individual genes. We know what it might be all the genes do, even though it seems like lots of genes can do kind of multiple things, especially working in tandem with other genes. We're definitely still figuring out about that. But now you can get a 23andMe test and see what kind of genetic propensity you have to certain diseases. So as that's become cheaper and more accessible, we've added that into this kind of research too with psychology. So that's been really nice that way. All right, so with that, correlation again does not mean causation. So just because the two twins in my earlier class, they both had a crooked tooth, that doesn't necessarily mean that it's a causal relationship. It might suggest that it is, and then we can do more investigation to find out, is that specific feature genetic? Because it's biological, that one probably is. But again, you would need to determine that there's not other outside causes, that there are not other environmental causes leading to both of these things. So correlation does not mean causation. If something is a causal relationship, it will have a correlation too, but just because it's correlated does not mean that one is causing the other. All right, so let's talk about how genes and the environment can interact. We'll talk about these right now, and then we'll talk about epigenetics, which I think is one of the most fascinating ways that genes and environment interact. So a passive genotype environment correlation is when, let's use an example of a child, when a child is born into an environment that's already supportive of their genetic tendencies. So the child isn't doing anything, and the environment is already just passively supportive of their genetic tendencies. So honestly, the ability to read, your speed at reading, your comprehension at reading is probably not genetic mostly, but let's just pretend it is. So let's pretend that your reading speed, your reading comprehension is 100% genetic. So these parents, let's say these parents have that genetic tendency to read really well, comprehend their reading. Likely they will have a house with more books. They might read to their child a little earlier. So that child also has that genetic tendency to read faster or read well. They're in an environment already that supports that. Versus maybe these children have the genetic propensity to do well at music. Well, more likely will be their environment has the opportunity for more music. So that's the passive environment. The child is not doing anything. The environment's already set up in a way that's supportive of their traits. So evocative genotype environment correlations is when, based on the child's actions and behavior, it elicits something from the environment. So a child who, again, these are just examples. These aren't exclusively genetically tied capacities, but let's pretend that being friendly and outgoing versus being less outgoing and maybe less friendly, let's pretend those are exclusively genetic. So if that was the case, we might see for a child who had the genetic tendency to be more outgoing, to be more friendly, other people will probably pick up on that, and this person will just develop more friendships. They will probably have a bigger group of friends, more people might like them, that kind of thing. And so the child, just because of their general behavior, because of these genetic traits, the environment around them has changed. So they didn't actively change the environment like intentionally, but the environment around them changed. Their behaviors, their genes have evoked a specific environment. That's what evocative genotype environment correlations are. Then active genotype environment correlations is one step further than that. The child actually chooses, based on the capacities they have, they choose or actively change the environment in a way that's supportive of their traits. So let's say the ability to do parkour is completely genetic. Again, probably not, but let's just use it as an example. So the ability to do parkour well, or free running, or running and jumping and things really well, let's say that's all genetic. That individual will probably choose maybe activities, clubs, sports that are interesting to them, that support those activities, support their genetic tendencies. So that's the active genotype environment correlation, where they pick, niche picking is that child is picking these areas that are supportive of their tendencies. So that's one way that heredity and environment can relate to each other. They have the genetic tendencies. Is the environment already supportive of these tendencies? Do they then elicit the supportive environment, or do they pick a supportive environment? The other way that environment and genes interact is through epigenetics, which is a really cool, well, I think it's really cool, a really cool feature of genetics. So again, going back to our discussion of what genes are, so we have our DNA strand, and if you took two clones, if you cloned yourself and you had a clone, the DNA sequence, the DNA structure, like the ATCG, would be exactly the same between you and your clone. But the epigenetics might be different. So for an example, a baby would inherit genes based on the prenatal development. Maybe there's some teratogens or some toxins in the environment, or maybe there's not. That might affect the way genes are expressed in facial features in the case of fetal alcohol syndrome, or brain development might change based on that kind of toxin in the environment. So the environment has affected the way the genes are expressed. So again, that DNA stays the same, so the ATCG, but the way the protein reads it might change. So epigenetic is about the changing the molecular structure of the DNA in the way that it might be more tightly wound, the DNA might be more loosely wound. The order stays the same, the ATCG, that all stays the same. But when it's wound around those proteins, it might be a little more tight. So when other protein comes to read it, it might not get the same message as if it was a little looser wound. So like literally the physical chemical process can change that. And so then that protein then goes and does a different direction in the cell, which could then lead to different behavior, different structures functioning, things like different hair colors maybe. So this is another really interesting example of a bidirectional interaction. Your environment can change the way your genes are expressed. So I've talked a lot about this, but I think it's a lot more helpful. Your book has some examples too, but I really like these two videos to explain it. So we're going to watch this whole one, and then maybe some of this example that relates epigenetics to Pokemon evolution. But let's watch this one, and hopefully it gives you a lot clearer examples of what I'm talking about with epigenetics.
say you have a clone or a secret identical twin, because you know that a twin or a clone is a genetically identical person to you, which is crazy, but it's true. Anyway, so you have this clone or your twin or what have you, and your parents decide that it's too much trouble to keep both of you. And so they give your clone to a kind of sketchy traveling circus. Now, I know your parents probably wouldn't actually do this, but just roll with me for a minute. So for the purposes of this example, let's just say that you grew up living a normal suburban American life, while your clone grew up with less stability and less access to nutritious food and less education, but more exercise and more access to sword swallowers and bearded ladies. And let's say you guys are 50 years old or whatever, you get together and have some lunch. So you're sitting there across the table from your clone. What do you think you see? Well, for starters, Clone probably has a tattoo on its face. And if Clone had stayed in the circus, Clone probably, professionally rides a unicycle over a tightrope, so Clone is probably in pretty good shape. But then again, Clone probably smokes cigarettes and probably was malnourished as a child, so might be slightly shorter than you. By the same token, let's say that you've been eating Sonic Tater Tots 5 days a week since 1992, so you're looking a little bit tubby. Plus, you spent the last 20 years at a really stressful job that puts you at risk for all kinds of weird health complications. So, at 50, you and Clone would probably look pretty different from one another. But it turns out, you don't just look different, you actually have become different. Now, if some scientists looked at your genetic code, they would say that your DNA was still exactly the same as that other person. That is to say, if your genome is a paragraph, all of the letters would be in the exact same order. But on another level, there would be a huge difference. And if we extend this metaphor, we can say that the letters are in the same order, but the spaces and the punctuation are all in different places. Of course, completely, potentially changing the message of that paragraph. The study of this genetic punctuation that I'm talking about is called epigenetics, which literally means above genetics. The epigenome doesn't change your DNA, but it decides how much or whether some genes are expressed in different cells in your body. Epigenetics looks at what happens to your genes over the course of your life and whether those changes could be passed down to your children or even your grandchildren. So here's the way epigenetics works. You have billions of cells in your body, and they each contain your DNA, the same exact blueprint of your genetic code. But just because they have the DNA doesn't mean that they know what to do with it. They need outside instruction from these little carbon and hydrogen compounds called methyl groups. The way these methyl groups control the genome is by binding to a gene and saying, do not express this gene. The methyl groups bind differently to your genome in a skin cell versus, say, a tongue cell or an eyeball cell. And that is one of the ways that a cell knows that, hey, I'm a skin cell, or hey, I'm an eyeball cell, or hey, I'm a muscle cell. In addition to methyl groups, epigenetics is also controlled by histones, which are proteins that are basically the spools that DNA winds itself around. Histones can change how tightly or loosely the DNA is wound around them. If they're more loosely wound, the genes can express more, and if they're more tightly wound, then they express less. So whereas the methyl groups are more like a switch, the histones are more like a knob. Every cell in your body has a distinct methylation and histone pattern. And that is what gives every cell its marching orders. Think of your genome, the DNA, as the actual hardware of the computer, while the epigenome is more like the software, which tells the hardware what to do. The genome is what's going to be doing all the work, but the epigenome is going to be telling it what to do. So the hardware of your DNA is going to be the same throughout your entire life. But these epigenetic tags do change throughout your life, and they decide what genes get expressed or not. Now, epigenetic information in a cell isn't permanent. It can change throughout your life, and it can be hereditary, and it can change over time, especially when your body is going through a lot of changes, like, say, during puberty. A bunch of methyl groups kick in and they're like, okay, so you guys over here, you're going to have to start growing hair, and you guys over here, I really need you guys to get behind giving this guy some really horrible acne. Or like when you get pregnant, which hopefully I won't, but when you do, your epigenome has to be like, all right, guys, everything's about to get a whole lot bigger in here, and we're going to have to pass something the size of like a Merriam-Webster's dictionary through that little pipe over there, so let's get this thing done. But it's not just these dramatic times when the epigenome is changing. It changes subtly throughout our entire lives, and it changes based on a lot of environmental factors, like what we do, what we eat, what we smoke, and how stressed out we are on a daily basis. Scientists have found that things like a bad diet can actually lead to methyl groups binding to the wrong place and making mistakes. And with those bad instructions, cells become abnormal and become a disease. And then basically all hell breaks loose and you get cancer or something. Epigenetics is a very young science, though we've known about the epigenome since the 1970s. It's only in the last 20 years that we've even known what effect these epigenetic tags are having on our DNA. And even after they got all that business straight, scientists still thought that all of our epigenetic tags were stripped off of our genome before they were passed on to our children. So if you started smoking 10 packs a day when you were 10 years old, that would certainly be a horrible health decision for you, but you wouldn't necessarily be harming your unborn children in any measurable way. However, the thinking on that is changing pretty rapidly, because it's true that a lot, even most of the epigenetic information from a parent is stripped off of the embryo's genome in the first few days, and fresh ones are created specifically for this new person. However, some of these tags get stuck on the genome and are passed down from generation to generation. And it just so happens that the more they study this, the more it looks like bad epigenetic information is being passed from generation to generation. And this is a whole new way to think about how we pass information between generations. Your grandmother was making dietary decisions that affect you today. As we experience all of these new strange epidemics, diabetes, autoimmune disorders, cancers that weren't appearing in previous generations, it's starting to look like these may be caused by epigenetic information passed down from our parents. I know, it's such an unbelievable buzzkill. There is no point in our lives when we can do anything without guilt anymore. The discovery that the environmental factors of parent experiences can be passed down from generation to generation was sparked in the 1980s. And this happened when some scientists were looking at the birth and death records of some people who lived in 19th century Sweden. As is a weird place to find a genetic revolution, but there it is. It wasn't just any place in Sweden. It was in Norrbotten, which is the northernmost county in Sweden, which is literally in the Arctic Circle. And despite the fact that Norrbotten was literally the worst place you could possibly choose to live in Sweden, there were some people living there in the 1800s. And they were completely cut off from the rest of the world. Let me clarify. These people were isolated. Like, if they didn't have a good crop year, people died. No, it wasn't ideal, but, you know, they didn't ask me my opinion, so I'm not giving it. So anyway, these people subsisted entirely on what they grew and the animals that they raised. And like I said, sometimes they starved, but sometimes they had huge, bountiful years of plenty. And when it happened, people totally went ape crackers. I mean, of course they did, because they were so freaking hungry, and then all of a sudden there was all this damn food everywhere. Anyway, there was a public health specialist who was looking at the effects of the people who grew up in the really bad starving periods of time versus the people who grew up in the eat all you can at the smorgasbord years. And you might already be guessing what they found out. People who went from relatively slim pickings to feed your face until you have to barf and then do it all over again in a single season, those people died an average of 6 years sooner than their starved out counterparts. And you know what sucks? So do their kids. And so do their kids' kids. Boo epigenetics. So, of course, now we all know, and we're all going to stop doing unhealthy things starting today. Unless the damage is already done. The damage is almost certainly already done. But hey, epigenetics brings good tidings along with the bad ones. For instance, we now know that certain types of cancer are caused by misplaced epigenetic tags, and scientists are now developing drugs that can silence the bad genes that were supposed to be turned off in the first place. Additionally, until recently, we thought that genes were the end-all and be-all of who you got to be. They were your blueprint, and you couldn't escape them. This outlook is not just kind of depressing, it also leads to a yucky sort of social prejudice. Because when you look at data without considering social and epigenetic factors, it might look like people with less money are less intelligent. So just like 15 years ago, there were scientists saying things in public, mind you, like, some people just have good genes for intelligence, and it just so happens that the poor people don't. Oh, it's so sad. Too bad, poor people. But you have bad genes. Well, it turns out that that is not even a little bit true. Not only are there a huge variety of social factors that affect how well people do on intelligence tests, but a genetic trait is also not just a product of genes, it's also a product of environment. Any one person's genome is determined by any number of decisions made by any number of their ancestors. And right now, you are making decisions that are going to affect people who are alive long after you are dead. No pressure or anything. And so I'm glad you've watched this, because when you and your clone are in your 60s and you sit down, have lunch, and he brings his son and his son has a tattoo on his face, he did not inherit that from you. This is Hank Green for The SciShow.
So hopefully those examples in that video made it a lot more clear of what epigenetics is actually doing. So again, just to kind of reiterate and explain it again. So the genes, the genetic structure, the genetic code, the order of the blueprint, the ATCG, that all stays the same. Your clone, it stays the same. An identical twin, it stays the same. But based on what you're doing and your environment, the things you're putting into your body, stress levels, things like that, certain genes might be read by a protein slightly differently than they would otherwise, which can lead to different traits being apparent. So let's watch another short video that shows another couple specific examples that I think makes this pretty clear. And rather than watching all of this, let's jump forward to here. And this is where a couple clear examples are of how epigenetics can change human and some other animals' physical features as well as behaviors.
So, should I be worried about Steph's relationship with the milk? But Steph, the day you were born, but how you look is depending on which ones are turned on and off. Your epigenetics. Right now, my baby Oliver has blonde hair, but Steph and I have brown hair. So, should I be worried about Steph's relationship with the milkman? Is Steph cheating on me? Of course not. At least, I don't think so. Steph! Steph, are you cheating on me? Oh, yeah, sure, that's what you would say. Now, what's really going on here is that right now, most of the genes in Oliver's system that produce melanin, which turns your hair brown, are turned off. Like, way off. Just like I had when I was his age. But when he gets older, those genes get activated, and his hair will turn brown just like ours. His physical traits will change as his genes turn on and off.
So again, this is dependent on the methylation and the histone pattern of each of your genes. So some of these things change just naturally as we age, but some of them are activated or deactivated or adjusted based on other factors in the environment.
Now, with animals, what epigenetic researchers have discovered is that some animals will be born with one set of genes, but potentially have parts of their DNA that can be unlocked by certain triggers in their environment. A great example of this are honeybees. Yeah, I know, I just dedicated an entire episode to bee talk as it relates to Peachette. Maybe I'm just going through bee movie meme withdrawals, I don't know. Bees are fascinating creatures, and in this case, they are the perfect EV example. So, like I covered back in that episode, female bee larvae can grow into either a queen bee or a worker bee. What makes the difference here is the food that they're given at birth. Bees given royal jelly become a queen, while everyone else becomes a worker. So in this case, royal jelly is basically the evolutionary stone for bees, determining what form the larval bee evolves into. It even changes the bee physically, where in their adult stage, queen bees and worker bees look way different, but in their larval stages, they're practically identical. It's exactly like two different evolutions that wind up with two different appearances despite starting with the same looking EVs. Stage.
So jumping back to this picture, so that changed just at a larval stage. In this example, the bees are essentially siblings, right? But we could clone a bee and have the two larvae be exactly genetically identical and then give one royal jelly and one not. And the one that got the royal jelly, because of that, because of the specific nutrients and things, it changes the way the genes are read and expressed. That leads to physical changes as well as behavioral changes in the queen bee versus a worker bee. So just that simple thing, or seemingly simple thing, of the different diet can change the bee that dramatically. There's another example that it will cover in just a second too about mice, which I think shows a really good example of the slight change in variation, where you can have a whole spectrum or range.
Queen bees and worker bees look way different, but in their larval stages, they're practically identical. It's exactly like two different evolutions that wind up with two different appearances despite starting with the same looking EVs. The queen and worker bees didn't start off as genetically different, but differences in their environment, the royal jelly, or lack thereof, led to wildly different gene expressions. And bees aren't the only place in nature where this happens. Another EV-like example happens in the agouti mouse, which has a gene which changes the fur of its coat. These two mice are genetically very similar at birth, but due to differences in nutrition, they have very different gene expressions, with one having a gene expression that causes it to become obese and have lighter fur. Same DNA sequence, different phenotypes, all due to environmental factors. Very different gene expressions, with one having a gene expression that causes...
So this is a really good picture and a really good example, I think, of showing how that winding around of the protein, that histones, the histones changes the genetic expression. So here, each of these mice are actually siblings in this study, But you could do this again with cloned mice. The reason they didn't is just because it's more expensive to then clone a mouse and then do this experiment. So these were all siblings. So genetically really similar. Just in general nature, most mice of this type will be about this color. And then in captivity, often they're closer to this color because of the nutrition that they're getting. The higher fat content, the higher calorie content, different vitamins and whatnot leads the gene to be wound either more tightly or more loosely. And that's why you see these different mice who got slightly different diets with a coat and size difference that is on the spectrum. So you've got the very small, very dark mouse, very large, very yellow mouse, and a whole range between them because that gene was wound more tightly versus less tightly. So as the protein goes to read it, it might be missing, like if it's supposed to be ACTG, it might be reading ATG. Or if it's really tightly wound, it might read it in order AG. And so then it takes that message to then change the cells and changes the colors. That's why you see some that are very different colors than others and this whole range between them. So that is what we're talking about in relation to epigenetics. That's what epigenetics can do. Essentially, with epigenetics, you can have a genetic trait, a genetic tendency that then is expressed differently later on in life, or slightly differently, or even dramatically differently. That can affect physical features or also emotional or psychological, cognitive features, processing speed, things like that. And so we're learning more and more of how many different things epigenetics can affect. So your blueprint might say for any given trait, maybe a personality trait like being outgoing. Maybe this is not at all outgoing and this is very outgoing. Maybe your genetic trait starts you off here in this developmental cascade. And then your environment is more supportive of being outgoing. And so there's a few more methyl groups that are more activated. Maybe your diet changes that too. I'm just using this as an example. And so it leads to you ending up, even though genetically you might have been over here, because of your epigenetics and other environmental factors, you might end up over here in outgoingness. So another clear example from your book that I'll jump back to really quick. Oh, that's forward, not back. Is the propensity for depression on this specific gene. It only activates when a person has a stressful life. So again, two clones or two twins might have that same gene, but if one of them has a stressful life versus one doesn't, the one with the stressful life would have that propensity for depression activated. So can do a lot of different things. So I think it's a really interesting newer area of research that you've probably not heard about. If you have, awesome. If you want to learn more or see if other aspects might be related to epigenetics, definitely go and look it up as a question paper, read some research on it. Probably next time at the beginning of class, I'll talk about how to find research and how to start working on a question paper if you're starting to get some ideas that are interesting to you. So next we're going to jump into prenatal development in chapter 2, but before that, let's take a quick break. Let's be back here at 10:57. If you have any questions about epigenetics, put them in the chat, or you can also just raise your hand at the end of the break too. But yeah, during the chat, let's be back at, let's say 10:57, 10:58, around there. All right, and we are back. So hopefully everybody is filing back in to class now after break. So we will get started back into prenatal development, and hopefully we can get mostly finished with chapter 2 before the end of class in just a little while. So prenatal development is split up into a couple different periods. Let me make sure that I'm still recording. Yep, and sharing screen. Perfect. All right, so prenatal development is split into a couple different sections, and it's even further split down, and your book goes into more details about the little splits. But we're going to focus on these three as big categories. So germinal period, embryonic period, and fetal period of development. So the germinal period is just the first 2 weeks after conception. So oftentimes the pregnant person doesn't know they're pregnant at this point. But essentially what's happening is the fertilization of the egg, which is then called a zygote, and then cell division starts to begin even before it gets implanted into the uterine wall. So from that fertilization to when it's implanted into the uterine wall, that is the germinal period. And you can see here it's fertilized and then starts cell division. And so now that zygote is implanted, that is the germinal period. So the next period is a little bit longer. It's called the embryonic period. So here it's now an embryo. You start to see organs beginning to develop. The one that we're generally most interested in psychology is the brain. So here we've got the forebrains beginning to develop, the midbrain and the hindbrain, and the spinal cord as well. You can also see things like stomachs and eyeballs beginning to develop during this time. And since these major organs are starting to form in between this second week and 8-week period, this is one of the biggest times that the embryo is susceptible to teratogens. So we'll talk about teratogens in a couple minutes, but essentially it's chemicals in the environment that can have an effect, generally a negative effect on the development of the embryo and fetus. So an example is alcohol. Alcohol in the environment can really harm some of these developing organs like the brain, the face, things like that. So the next period is the fetal period, and this is essentially the whole rest of pregnancy. So from that second month to the last month of pregnancy. So your book again breaks it down into even more detail than we're going on here, and that can be really interesting, especially if you are pregnant, you have a family member that's pregnant, or you've been pregnant before, looking at the time periods. So right now, for example, my brother, his wife is pregnant, and I think she's somewhere in between this space, in between 28 and 32 weeks. So that fetus at this point probably has iron starting to be stored in the liver, is about this long, versus when they first told us the embryo was only about the size of like a sprinkle that you would put on a donut or something. So pretty neat to see. I remember we had an app for our children that would tell us like the growth. So now the baby's about the size of an orange or a pear or something like that. So that's always fun to look at. But that's the fetal period. So the teratogens generally can have a bigger detrimental impact during the embryonic period than the fetal period, but there's still a lot happening during the fetal period. So let's specifically look at the development of the brain during the whole prenatal development. So if you can see, hopefully you can see with the screen sharing, with it capturing in high enough quality, because the picture itself isn't super high quality. But here, this is the brain at 29 days versus 52. I can't even read that one. But this one is 70. This one is 20 weeks, 6 months. So still prenatal here. This is 9 months, about when a baby is generally born. So you can see the change, the dramatic change in the brain during the prenatal period versus when a baby is born. When you have an infant, the brain is very similar in appearance and structure as it is to an adult brain. So you might notice, oh, it's not quite as wrinkly, it's not quite as big, and that's about the differences versus in those 9 months, it goes from a couple cells to all these billions of brain cells in their proper place. So at week 5 is essentially when a ton of new neurons start to be developed. So essentially stem cells at that time, at about five weeks, start to specialize into neurons. And there's other neurons. There's other cells in the brain as well that are kind of like, you could think of it as like a scaffolding for the neurons. So they kind of follow this scaffolding to their final place, like where that neuron will live. So that stem cell, as it specializes into a neuron, it kind of migrates to where it's going to end up. So whether it's in the rear of your brain, the front of your brain, this part of your brain, the very middle, it kind of the stem cell specializes and then moves to where it goes, and then it stops there, and once enough brain cells get around in the area, it starts making connections. So at about this point in time, you've got a good portion. So at about 20 weeks, you've got a good portion of neurons already in the place that they should be, and they start to reach out axons and dendrites to communicate with other neurons. So that neuron, so you start making a ton of neurons at about 5 weeks, and then between about weeks 23 to 25, depending on the individual, you start making connections between neurons, and you can see the first evidence of messages being sent back and forth between neurons, which I just think is fascinating. You obviously can't interview a 25-week-old fetus to see what kind of sensations or perceptions that they have at that point in time, but I just think that's a fascinating thing. Like, the brain starts lighting up at that point. So brain cells are growing, developing, moving to where they go, then they reach out, and then they start talking to each other. So it's like little sparks here and there, and then it's all working together really dramatically quickly, honestly. That's happening at 25 weeks, and then by the end of 9 months, you've got most of the brain cells you will ever have in your life. It's just kind of fascinating to me. But also goes to show how these kind of teratogens can really impact brain development. So you continue to develop neurons even up to about age 2. So at about age 2, those stem cells basically stop specializing into neurons. And at about age 2, you have essentially all the neurons you will ever have in your brain. With that being said, it does look like there might be a couple places in the brain that we might be able to still develop new brain cells. Brain cells don't develop the same way as some cells do, like through just splitting off into 2 new cells. It has to be a stem cell specializing to a neuron. So in the hippocampus, it does look like we might still, even into adulthood, be creating new brain cells in that specific area. And then maybe one area of where we're processing smells as well, but that one's not clear. But in the hippocampus, it seems like we do. But it's not a massive number of new cells, not like it is when we're at that 5-week period and there's tons and tons, and by tons, I mean hundreds of thousands to billions of cells being created quickly. So at about age 2, you have essentially all the brain cells you will ever have in your entire life. And at that point, you do a lot of pruning of connections. So up until age 2, you're still making a lot of connections. You can imagine each cell splitting off more axon branches, a lot more dendrites to talk to all the other neurons around it. So at age 2, whatever you're using, you'll keep. Whatever you're not using, you'll lose. So the neurons themselves will stay alive. But let's say a neuron has 4 dendrites to receive messages from other cells. So let's say I'll just use my 4 fingers. So my hand is kind of like the cell body. My thumb could be an axon, like sending a message off. So there's these 4 dendrites talking to other cells. But let's say I'm not using this specific neuron very much. So to conserve energy, it'll reabsorb some of those dendrites. So now I have 2 dendrites getting messages. It's taking less energy to run this cell, so the brain is happier. It's a little more energy efficient. So at about 2 years old, that pruning starts to happen a lot. And that process happens through our entire life. If we're not using aspects of the neuron, it might reabsorb some axon branches or some dendrites. Later on, if we start to use that portion of the brain and need more connections there, it'll make new connections. And the creation of a new dendrite branch can be really quick. Depending on how far it has to stretch in the brain, you can make a new connection somewhere in between like 12 and 48 hours, which is pretty dramatic. I want a picture of a neuron up to talk about that, but yeah. Yeah, one really fascinating, at least I think it's fascinating, it's slightly tangential here, aspect of that is with the brain plasticity that you can develop those new connections pretty quickly like that. If you ever start weightlifting, or maybe you lifted before but you haven't recently, if you ever start weightlifting, you'll notice that you will go up in weights really quickly at the beginning. So in the first couple weeks, to even first month or two, you'll go up really quickly in weights. But then you'll hit a plateau where you stop increasing weights so dramatically. And a lot of people actually, when they're starting to do weightlifting, they'll get disappointed. They'll have like a 5-pound or 10-pound or 20-pound jump to their weights and be really impressed with what they're doing and how much more they're lifting. But then it kind of stops. That actually is caused by connections in neurons. So your brain, since you're using those muscles more, it tells the neurons that send a message to your muscle. It says, hey, make new axon branches. Tell more muscle cells to activate. These muscles are being used more than they were in the past. Tell more muscle cells to work. So you're not actually, until about month 1, you're not really building new muscle cells yet, but you're getting new axon branches telling more muscle cells to activate. So it's a pretty interesting phenomenon that as you have that plasticity in your nerves to tell more muscle cells to activate, and it leads to you lifting heavier weights. But then once you have to catch up with the muscle cells actually growing and dividing and having more muscle cells, your growth and your going up in weight is a lot slower at that point. Same thing happens for different parts of your brain. If you are using the section of your brain for language development at age 2, if you're learning 3 languages, that area will be a lot more dense with connections than if you're only learning 1 language. If you then later on in life start to learn more languages, you'll make more connections in that area. So this also goes to the plasticity if you get a brain injury. So if you get a brain injury in, say, this area of your brain right here, the other areas of your brain and the other parts of your brain in that network can actually accommodate for that. They will take over the responsibility that was those specific cells. And that's some of the only ways brain cells die generally, is because they're not getting the proper nutrients they need, they're getting poisoned, they get damaged. Other than that, you basically have the same neurons from the time you are 2 years old to the time that you die, which is really interesting, I think. All right, so I kind of went on a couple tangents there. It took a little longer than I expected, so sorry about that. But are there any questions about brain development up to that point? Feel free to either raise your hand at any time in class. Feel free to raise your hand or type a comment in the meeting chat. All right, so we'll talk about some other aspects of the prenatal experience. One is infertility. So about 10 to 15% of people experience infertility. So that's a lot larger than I think we generally realize. So it's not something that's generally talked about, at least in the US, how common infertility is. And it can be caused by a lot of different things. So it's defined as the inability to have a child after 12 months of regular intercourse with no contraception. So that can be caused, though, by a lot of different things, like not ovulating or blocked fallopian tubes, too few sperm, or sperm that can't move adequately. So if you go to a doctor for this, they will start off with some of the most common kinds of treatments. So ovulating for one, to make sure that ovulation is happening. And if that doesn't solve the problem, if you're still not able to have a child, then they'll try more and more treatments, and that's the way they kind of diagnose and figure out exactly what it is. There's also other tests they can do. So it's very different for different individuals. I know some people who've had just quick hormone therapies for fertility, and it worked really well, versus some people have had to have surgery in my family, or even had to go to do in vitro fertilization. to be able to have a child. So it really just depends on what it is specifically with you and a partner that is leading to the infertility. So going back to teratogens, especially because we mentioned this, let's talk about a couple specifically. So essentially, a teratogen is anything, usually a chemical, that can cause a birth defect or negatively alter cognitive or behavioral outcomes. So the dose is important. Like under a certain dosage level, this chemical might not be harmful, but above a certain dosage level, it might be harmful. And the more it is, it might lead to worse outcomes. And then genetic susceptibility. So is that fetus susceptible to this specific teratogen? And also, what time in the development of the fetus is this happening? Is this happening at week 3 versus week 20? Depending on the chemical, might make a change at one of those times, but not one of those times. So alcohol is a really clear example of this. So fetal alcohol spectrum disorders are caused by the teratogen of alcohol. It can lead to facial deformities in the developing features. It can lead to defective limbs, face, heart. So it can also lead to learning problems and intellectual disabilities because the neurons are not exactly maybe where they should be or connected up how they should be or myelinating like they should be. So with that, 1 to 2 servings of beer or wine for just a few days a week could cause problems in the fetus. So the recommendation is not to use any alcohol when pregnant at all. And that's generally the recommendation for most of these teratogens that we're going to talk about. So caffeine is one that it's still not super clear, but the recommendation is use a different kind of stimulant. So talk to a doctor about anything that you're using. A good example of that is actually marijuana. It's used medicinally in a lot of states now in the US. So with that being said, for the pregnant person, using THC is probably not the best idea because it is related to low birth weight, likelihood of needing a NICU, stillbirth, and lower intelligence in children. So just like other medicines on here, opioids, for example, talking to a doctor about using THC is important so that you can find a suitable alternative that would be safe for the developing fetus. So there's other similar medications that can do the same thing that would be safe for the child. As an example, my wife was on a pain medication both when pregnant with one of our children that she had to switch to a different one so that the original one wouldn't harm the child. So yeah, just talk to the doctor. Even if you're using it recreationally, talk to a doctor so you can find a suitable alternative to help with the development of the fetus. Heroin and cocaine also have an additional problem that it can lead to addiction in the fetus, and so that can be really difficult as well. But yeah, we're not going to go into detail on all of these. Hopefully you did read them in the book. Another couple things that I definitely wanted to talk about was maternal diet. So again, talking about the environment for the fetus to develop in. So things like having obesity, increases rates of hypertension, diabetes, respiratory problems, not just in the pregnant person, but also in the fetus as well. So fetuses need a lot of folic acid, omega-3 fatty acids, proteins, vitamins, and many minerals like iron that an adult human doesn't need in the same amount. So things like prenatal vitamins can help with that. Generally, eating healthy foods can help with that. So trying to eat a change and have a more balanced diet is really good, which can be difficult because some people, when they're pregnant, they have certain cravings. Some people also have food aversions that they just don't want to eat certain foods. So trying your best to get in the proper nutrients to create a proper environment for the development of the fetus is really important. And then obviously keeping out the harmful chemicals like alcohol is really important as well. With that, in the last couple minutes, we can talk about how maternal age is another parental factor in a developing fetus that can matter. So there's 2 time periods that seem to be a higher risk for the developing fetus, and those are adolescence and people 35 years and older. So in adolescence, there tends to be a higher infant mortality rate. Prenatal care is often not as good, and you can think of the social cognitive factors that might go into that. Maybe a pregnant adolescent, maybe if they're pregnant at 14 or 15 or 16, doesn't want to tell people about it. So they don't go and seek a prenatal doctor. They're not taking prenatal vitamins. They might not realize or even know for a while that they are pregnant. So they're less likely to seek prenatal care, and that can lead to higher infant mortality rate, higher rates of things like Down syndrome as well. Similarly, at 35 years and older, it also leads to things like higher rates of Down syndrome, higher risk of low birth weight, preterm delivery, especially after getting past age 40. It's a lot higher likelihood. So like 35, it's a little more risky than say at maybe 25 or 30, but after age 40, the risk jumps up again. So increased miscarriage, increased preterm birth, stillbirth, and fetal growth problems. So with that being said, that should maybe suggest times to be trying to have children. And also, especially if an adolescent is pregnant, encouraging them and supporting them in getting the prenatal care that they need. Some other things like exposure to lead or radiation and some pesticides can cause abnormalities in sperm specifically. So it might lead to infertility and problems getting pregnant, can also lead to miscarriage or childhood cancer. So, and then also the relationship with the mother is very important as well. So partners that are more supportive, egalitarian, and positive with each other help care for the pregnant person better and lead to less stress, a more healthy environment for even the fetus to be developing in. So those levels of stress hormones, those are still in the same body for the developing fetus. So that can be another aspect to support a pregnant person as well. Prenatal care can look really different for each individual person who's pregnant. So there's a lot of different options. Usually, especially in the US, it involves regular doctor visits where you'll kind of be taught about what to expect, specific tests at different points in the life cycle of the fetus. So at a certain point, now an ultrasound would be done or specific blood tests. You might be doing some of these visits in groups or one-on-one meetings. Exercise for a person who's pregnant is really important. So keeping their own body healthy to keep the environment healthy for the fetus, and then a balanced diet like we mentioned before. So we'll talk about birth in the postpartum period, and that's basically the last thing for chapter 2. We'll talk about that next time. If I breezed over anything just too quickly and you still have questions about it, feel free to put them in the chat here, even after class is fine, or email me those questions as well. But we're now at the end of our class time, so thank you all for being here. Hopefully you learned some interesting things today. Hopefully you liked learning about epigenetics with our focus on that today too. And yeah, I will see you for the conclusion of chapter 2 and the beginning of chapter 3 on Tuesday. So thanks very much.